Hello and welcome to Science Research Weekly, Season 5, Episode 14, Not-so-Rarified Space. So get ready for chemical synthesis a la large language models, an amino acid for Alzheimer's pathology suppression, a trans-Neptunian object with an atmosphere, PhysForge versus ProtSpace, and career grants, cancer grants from the Department of War. This will cover materials from May 1st to May 7th. Let's get started with our research articles. Starting out here, we have the article Chemical Reasoning in LLMs, Unlock Strategy-Aware Synthesis Planning and Reaction Mechanism Elucidation. So, chemists were able to use computational tools, specifically large language models, to bridge the difficult issue of synthesis planning in sort of chemical steps, especially with mechanism to get, from one chemical to another, sort of complex formulations. And so using these large language model frameworks, they actually were able to get very good alignment with independent expert chemists. And they were able to find plausible reaction mechanisms from first principles. And that with higher model size generation, they were able to scale their chemical reasoning capabilities. So it looks like this is a useful framework in chemistry for large language models. All right, moving on, we have oral administration of arginine suppresses A beta pathology in animal models of Alzheimer's disease. So A beta or amyloid beta, that aggregation was suppressed both in vitro and in vivo. I believe the model was fruit flies, the animal model. And so arginine, that's actually an amino acid, a very common amino acid. And additionally, oral administration suppressed the amyloid beta pathology and its toxicity. And so arginine might actually be a safe and very cost-effective drug candidate for dealing with Alzheimer's disease. So it seemed like a very simple thing. And arginine is not a complicated molecule. It's pretty straightforward. So hopefully this bears out and this can be used effectively. Next up, we have multiple independent genetic code reassignments of the UAG stop codon in phyllopharyngean ciliates. So most organisms, 20 standard amino acids. So we just talked about amino acids earlier. But so with amino acids in most organisms, there's 20 standard ones that are coded by codon. So those are a set of 3 nucleotides. And then they also have 3 stop codons. So in the RNA, UAA, UAG, and UGA, those terminate translation. They're stop codons. They don't code for amino acid. However, in some ciliates, it turns out that they have reassigned the UAG stop codon to code for, or at least putatively code for, leucine, another amino acid. So it shows that, while sort of the fundamental dogma of biology, DNA to RNA to proteins, and how that second part, that translation, the RNA to proteins, is fairly conserved. Standard amino acids with 61 sense codons and 3 stop codons isn't universal. Sometimes the stop codons can be co-opted to something else, shifted back to something else. All right. We have the next article, detection of an atmosphere on a trans-Neptunian object beyond Pluto. So the object sorted into 2002 XV93. It's a planetoid, maybe a minor planet, you maybe call it. It's 250-kilometer radius, and with its refractive signature observed, there's an indication that it actually has a very thin atmosphere, as opposed to having no atmosphere like all the other sort of planetoids or objects out there. So this is definitely a surprising finding. And the authors suggest that maybe how it got the atmosphere is through cryovolcanic activity or even an impact from a small icy object. That it's not clear how an atmosphere would be sustained given how we know atmospheres work with planets or planetoids. So there's definitely growing our knowledge in that area. Final article here, bioprinting of microtissues within mechanically tunable support baths to engineer anisotropic musculoskeletal tissues. So normally in cells developing into tissues, they have a lot of these constraints, mechanical constraints by surrounding tissues. And so when you get to bioprinting, you print out cells into living grafts, etc., to sort of make your own tissues from cells. You might need to also build in those mechanical constraints to get the type of tissues or microtissues that you want. And so that's what this paper did here. They have these support baths that actually have mechanical properties that you can tune to get sort of post-printing mechanical stresses in the right way to form the type of microtissues you want that better mimic actual tissue formation. All right, that is the end of our normal articles. Let's turn to a couple preprints. The first one from Archive, PhysForge, generating physics-grounded 3D assets for interactive virtual world. So PhysForge is a decoupled two-stage framework supported by PhysDB, a large-scale database with over 100,000 assets with 4-tier physical annotations. And so, yeah, they're able to create these 3D assets that have physics-based constraints for use in virtual or AI, etc. applications. All right. Second preprint from bioRxiv, ProtSpace, Protein University in your browser. So ProtSpace, it's a free open source web application to visualize protein language model embeddings as interactive maps. It zero installation, so you can run in your browser. It takes annotation types from different sources like Uniprot, NCBI taxonomy, etc. And so that might be of use for you if you're doing research on proteins. With that, we will close our time here with some grants. I was not able to see any of the new grant packages. I was having an issue with my code. Hopefully that's fixed next week, and I didn't see any tools that were of use for this week. So with that, whole dump of Department of War grants for cancer and other diseases. I just took a representative one, the Cancer Idea Award. And so they support innovative, untested, high-risk, potentially high-reward concepts, theories, paradigms, and/or basic cancer research. And then, of course, this is just one grant. There's ones for more specific cancers or even things like Alzheimer's disease. So you can check that out. And then finally, we have from the National Institutes of Health, the Bioengineering Research Grants. These are BRG, R01 clinical trial optional. And this funding encourages collaborations between life and physical sciences. So that's what that bioengineering comes in. With that, thanks for joining me for Science Research Weekly, Science On.